Welcome everyone to this webinar on gene therapy organized by Genopole and Choose Paris Region. So thank you for being with us today. I am Romain Vallée, health expert in Choose Paris Region, and our agency supports international companies willing to set up and expand in Paris Region. So I will make a very short introduction on Paris Region. If you have any question, you can use the chat function and we will answer them after the presentation. So some quick figures about Paris region. We are 12 million inhabitants and have more than 1 million companies, including 8,000 startups, which makes our region very dynamic and innovative. And Paris region is the first world destination for foreign investment in R&D, and we have also the highest concentration of qualified students in Europe. We have major clusters in health, and just a word on two of them. Medicine, Paris region, which is a global hub for health innovative technologies, and Genopole, the first biocluster in France, which has a very strong scientific and business campus. And in a few minutes, a short presentation on Genopole will be done by Laurent. So regarding international rankings in the field of health, we are the first region in Europe for the number of companies in the pharmaceutical sector, and the second in Europe for the number of companies in the field of medical devices. We also have the chance to have the largest hospital network with 8 PHP. That is the first for clinical research in Europe, and have more than 8 million patients annually coming to this public hospital network. And collaboration with private host companies, small and large companies, are frequent. On research, the region features a high concentration of world-famous academic research institutions in the field of health, and we have more than 60 biotech research centers. Today, you will see, we'll have the chance today to have with us Galy from Inserm and Genopole. This is not well known, I think, and it should be better known. The fact that doing research in France is very competitive, thanks to the incentives we have, like the tax credit system and R&D subsidies. And you can see on this slide the research cost differential between France and some countries. So if you are interested in your business, in setting up your business in Paris region, Choose Paris Region Agency and Genopole can help you on the soft landing with all these aspects, from business plan finalization, partner search, and registry of your company to welcome you in your region, in our region. And last point, Paris is a great place to both live and work, and I'm not the only one to say it. So as we often say, you could come for business and stay for life. Thank you for your attention. And now let me introduce Laurent from Genopole.
Okay, I'm Laurent from Genopole. Thank you, Romain. So let me talk a little bit about Genopole. Genopole is a French biocluster, the leading biocluster in France. So we can talk about to do business in France. France is not only cheese and wine, but it's also business and biotech. We are, as Romain said, we are part of the Paris region. We are a gateway to go to Europe. We can go everywhere in Europe very quickly, by train, by plane also, even if now it's not so easy. And what is very interesting, we are at 30 kilometers from the Eiffel Tower, and of course we have, what is very interesting for a company to be located at Genopole is that we have a UPS hub next to Genopole, which means that the location is pretty good for company, for the Paris region. Something else to add, and for your information, what is very interesting in France is a R&D tax system credit that we have in France, which removes 30% of our tax, which makes us a very competitive country when you are doing R&D in biotech especially, but in every kind of field, of course, for France. Genopole, in some words, in three keywords, we are the French leading biocluster. I told you we have been created in '98. We are working in any kind of biotech, which can be medical, industrial, biotech, environmental, agricultural, or marine, and we are working with a team of what we call a JIP, which is a non-for-profit organization. We are working on an ecosystem to help companies to develop on an ecosystem, a friendly ecosystem for companies here at Genopole. Some key figures about Genopole. So we have currently 83 companies, from startup, from 1 people to 50 people, working on any kind of biotech, I told you. We have 17 academic laboratories. You will see tonight two labs talk about what they are doing in gene therapy. We have, of course, 25 platforms, which are technical platforms, which are very interesting, which means that companies, they don't need to invest in platform, technological platform. They can use our platform. And we are, of course, raised a lot of money on this campus for 14 years. And we are more than 2,000 people working at Genopole. So let's see what is Genopole. We are a biocluster, but also a biopark in the same times, if you want. We have, as you can see, a lot of space for companies. So we have these 83 companies on the campus. We have 1 incubator building for startups, for small startups. We have 1 hospital of 1,000 beds just down the road. So it's pretty interesting to get collaboration with hospital. We have a bioproduction of gene therapy, clinical gene therapy, so it's Yposkesi, and Mrs. Blondel will talk about that. We have also 1 at Cytopolis, you can see on this side, with Geneton and INSERM teams are working on cellular therapy. And Dr. Galy will talk about that just after me. Some companies that we have on the campus, you can see we have American company, which is like Illumina, which are the leader in sequencing. We have also New England Biolabs, a leader also in enzyme. We have many kind of companies. We have also Santen, Japanese company. We have Farming, a Dutch company, working in different area. Okay, most of our area are on health, but also we have a lot of in environment, in industrial biotech, in agri-tech, but also in food tech. And we can notice that we have Insect, which just raised $200 million last week, in fact, and we are pretty proud about this company on Genopole. So finally, what I can say about Genopole, it's a continuum of biotech. We have academic labs, we have biotech company, we have a platform, shared technology platform, and university, everything on the same area. So thank you very much. So as I told you, you can contact me later on. And let me introduce you to Galy, which will talk about gene therapy.
Hello, everyone. Thank you for this invitation. I'm going to be talking to you about gene therapy scientific advances and therapeutic applications. I have been here at Genopole for about 20 years, where I was recruited from the US with a startup package. And I'm working currently at Geneton as the head of the academic research, and also started a new laboratory in Genopole, which is an accelerator of technological research. So the environment here in the south of Paris is a little hub in gene therapy. It's actually a very important hub. It's a hub that was, in fact, initiated by the vision of the French Muscular Dystrophy Association, the AFM Téléthon, to start up a strategy to cure genetic diseases with gene therapy. And it's a lab of Geneton, which is now 30 years old, has been spearheading this effort. This has created spin-offs such as GenoSafe, which is a CRO that specializes in gene therapy quality control, as well as Yposkesi, that you will hear about a little bit later, that is a CDMO in gene therapy. But there are other actors such as INSERM Laboratories, this accelerator of research, which is combined with another cell and gene therapy lab, as well as other companies like Texcell, and also newcomers in the field. You'll be hearing about White Lab Genomics, which is a small company that is also interested in gene therapy on a different angle. So gene therapy as a whole is really a new way to treat diseases with gene-based medicinal drugs. It is about 1,300 plus ongoing trials worldwide, and that map you're seeing here is in fact a recent caption from clinicaltrials.gov that shows the complete dissemination of efforts in gene therapy all over the world, of course with a efforts more intense in the US and in Europe, but also very, very strongly in Asia. 60% of gene therapy trials are in cancer, and there's already 9 approved drugs in Europe, a little bit more if you count in different ways, but this is a very, very dynamic biotech and pharma sector. And as you can see here on this graph, and you can see some illustrations of some of the gene therapy drugs, there's a huge increase in the number of applications, for instance, in the US at the FDA, following the approval of some of the gene therapy indication, some of the gene therapy drugs, sorry. And these gene therapy drugs can be very different. They can be cells that are genetically modified. They can be also vectors. They can be different things. So it's a very rich field. So gene therapy addresses unmet medical needs of many rare diseases, not all, but there's between 5,000 and 6,000 different kinds of rare diseases, which are mostly of genetic origin. And that concerns, of course, for every disease, very few people, but collectively it's 30 million Europeans. So it's very important. And 95% of these diseases have no treatment. So gene therapy can address unmet medical needs in many of these rare genetic diseases. And in fact, I'll give you a couple of examples. One is a disease that we're working on at Genopole that we have, in fact, that I have had the privilege to lead with a number of colleagues, both at Genopole and, of course, outside in London, as well as in Paris, but also in the US, where we've, in fact, collectively worked together, in fact, also helped by the European Commission. I was piloting this Net4CGD international network to try to develop a gene therapy for X-linked chronic granulomatous disease. And this has been a successful clinical trial, where you can see here one of the patients treated in Boston. And it was recently published in Nature Medicine. And this treatment, in fact, even though we don't understand every aspect of gene therapy in this disease, which is complicated, it still profits and benefits many of the patients. So one of the companies actually is trying to, in fact, develop this product and take it further, perhaps, to a marketable drug, which is a huge promise for the patients. Another disease to which Genopole was initially associated but has been developed mostly in the US is the gene therapy of spinal muscular atrophy, which is done by one single injection of an AAV vector. And pilot and initial trials were done in Columbus, Ohio by Jerry Mendel, and they have led to spectacular results in these very severely affected infants, which can then, after just one single injection, start to develop when the dose of vector is sufficiently high, and that leads to at least a very, very improved development of their motor function. So now it's, in fact, an approved drug called Zolgen SMA, and it's a very impressive result in gene therapy. Now, gene therapy can also address unmet medical needs of many frequent diseases, not just rare diseases, but frequent diseases. And you've probably all heard about cancer immunotherapy with gene-modified lymphocytes, such as CAR T cells, but also tumor-specific TCR T cells. And there are other types of immune cells that can be readdressed and rearmed to try to fight tumor cells. There's also oncolytic viruses, which can be genetically modified and in fact directed against tumors. And there's in fact gene-modified viruses such as Imlygic that are in fact also approved drugs in this case, for instance, for melanoma. So you can also think of completely new applications of gene therapy, and many people, including ourselves at the Accelerator of Gene Therapy, are thinking of gene therapy as a new way to vaccinate. The old way is to bring pieces of the antigen, of the virus, of the microbe in the form of the microbe itself, or perhaps little pieces of it. Now you can, in fact, re-engineer these viruses into in fact, vectors. You can also put viral particles with you can also inject patients with DNA, and we're thinking of even more exotic ways to perhaps, in fact, bring the antibodies directly into the people. In the case, for instance, of vector immunoprophylaxis, or in our case, we're trying to reprogram B cells to re-engineer them to, in fact, recognize the microbes. So this can be very, very innovative. And in fact, indeed, gene therapy is very innovative. It's a complex and very evolutive type of technology, and many technological platforms are available for gene therapy. You can do gene therapy by injecting your products in vivo, for instance, intravenously, or in any kind of way you can administer that directly to the patient, or you can in fact gene modify cells ex vivo. for instance, CAR T cells. And we've seen also a really great evolution in the different technologies used to, in fact, genetically modify the cells. In the past, people were using plasmids that are not very efficient. This has moved to recombinant viral vectors in many applications. And even in that field, people have moved from the gamma retrovirus, for instance, which were causing problems by the insertion linked to LTR activation of neighboring genes. And now we're using self-inactivated viruses such as lentiviral vectors. People have moved also from adenovirus to AAV in the case of genetic disease, even though adenoviruses, of course, are great tools to vaccinate. And in fact, there are, for instance, many projects in the COVID vaccination field that are using adenovirus to try to vaccinate against COVID. And people are moving from gene transfer, where you add gene, it's a gene addition strategy, to genome editing, where you're going directly into the genome to, in fact, completely edit and modify what you want to modify. And I would like to really acknowledge the Nobel Prize in Chemistry that was just announced yesterday for the development of a method for genome editing to Jennifer Doudna and Emmanuel Charpentier, that I congratulate for this fabulous discovery that we are all using every day in our lab. And I want to emphasize, for instance, one of recent publication from our unit in Genetton, from one of my colleagues at INSERM, Mario Amendola, who has, for instance, engineered a way, for instance, to gene modify hematopoietic progenitor cells, CD34 positive cells, with CRISPR-Cas9 to insert very precisely a transgene into the globin locus. And in this way, when the cells differentiate into red blood cells, they're going to express whatever cargo he had put into these cells. So you can see here on that slide on the right, the production from progenitor cells of red blood cells that are green, because they are expressing GFP. And he has demonstrated that engineering stem cells for erythroid protein expression can be used to basically bring enzymes and treat many diseases ranging from hemophilia to various metabolic disorders. So gene therapy is not always easy. There are many challenges in gene therapy. One is, in fact, the use of gene therapy for systemic administration. And one of the big problems, for instance, is the induction of immune responses against the vector or the transgene. There are also other issues. But in this particular question, we have worked with many of my colleagues, as well as many other people in the world, to try to overcome, for instance, in the case of AAV administration, the immune responses against the capsid or the transgene. So we have worked on various strategies to be able to redose AAV, as we know that a single injection might not always be sufficient to treat patients. So there's very exciting new ways to try to overcome that problem, or to try also to overcome the problem of the seropositive people. So that means there are people who have already antibodies against the vector of gene therapy, and of course who cannot be administered. But there is a solution for that, including the use of peptidase that can cleave these antibodies and perhaps allow administration of the vectors even in seropositive individuals. There's another challenge, which is the manufacturing of clinical grade vectors in large amounts, sufficiently large to conduct clinical trials with many patients, or even beyond clinical trials to try to commercialize the vectors. So in many cases, not always, we're in fact doing transient transfection. So we take producer cells, put plasmids in there to in fact collect the particles that we want that are carrying the gene of interest. We have developed, and many labs are developing, of course, in pre-industrial or industrial processes to produce large-scale batches, and that is going all the way to industrial 200-liter scales and much, much beyond. And this is an absolute challenge to make all of this. It's very costly and very difficult. So we are recognizing all the need to integrate research with industrial development in cell and gene therapy, which is why we put together at the Accelerator of Research Technology and another laboratory, put ourselves together to in fact propose one of those industrial integrating center that is connecting between research lab and industrial partners to try to develop as quickly as possible this proof of concept and to promote early phase trial development. So thanks for your attention, and I have my coordinates here if you want to contact me. Thank you.
Hello everybody, just to introduce Mr. Julien Cottineau from White Lab Genomics, and he will talk about platform and for gene and cell therapy. Thank you, Julien.
Thank you, Laurent. So we are a team of 6 people with expertise and complementary skills in biology, computer sciences, and business. And as all of you may know, the gene and cell therapy industry today are facing many challenges, and some of them are listed here, and Anne has described some of them also. So, for example, improved target delivery, biosafety, or transgene expression. And at White Lab Genomics, what we want is to help you accelerate gene and cell therapy development using our AI-driven platform. So our technology is based on the graph knowledge database. So it can be described as a collection of items with specific properties which are linked together to create a network of information. So you may recognize some of the companies that's using already those technologies. And compared to classical databases, knowledge graph databases are showing higher performance and accuracy and are more adapted to machine learning. So to build our platform, we do aggregate medical and biological data. It could be documentation as publication, patents, or business filing. It could be also biological data as genomic, proteomic, or physiological information. And finally, we also developed our own gene and cell therapy dataset. Today, our platform is look like that. More than 2 million of nodes and more than 30 million of connections. And this is still growing. So to process and analyze our database, we develop our own algorithms. It could be natural language processing, machine learning to generate specific models. And we use also federating learning, working with our partner to reinforce the models specifically to their project. So let's take a real customer case. For example, how to find the best vector for a specific gene therapy. First, an observation. There is more than 1,000 capsid variation and more than 10,000 documentation that have been published into the literature. And so this quantity of information and data is overwhelming for a human perspective. So based on these needs, we developed with our platform a screening tool to benchmark the vectors and all the variation for the capsid. So let me show you a little demonstration of our interface. So first, we organize and structure all the information by cards. So as you can see on this dashboard, you will find all the categories specifically by cards. And you can put a specific query, for example, AVP1 protein, and so you can access to the specific publication, and all cards will be a specific publication with main information. And of course, you will have filters option to adapt your search. And if you click on the publication, you will find more details and information. So going back to other documentation, for example, patents or clinical trial, this is the same structure. Concerning more biological information, let's say genes, you will find a specific list corresponding to your query, and you can select directly a gene, find general information, associated serotypes and genes, a tool for variant alignment, and also tables that you can find directly the specific documentation for each variant, for example, publication or patents. But also you will find related information as transcribed related protein. Concerning the protein, you will find the same. So basically, from the query, we generate a list of proteins that are associated with this query. You will find general information, related serotypes, and similar protein. You will find also structure information, could be 3D structure, but also to map all the variation corresponding to the specific capsid. You will find all the variation based on the protein structure, and you will find also tables that summarize the information of those variations. And each publication specifically to one variation will be found directly here. The same for protein post-modification with a lollipops schema and tables that summarize the publication and the patents associated to this specific modification. So just to finish my presentation, I would like to say that at White Lab Genomics, we are really open to any use case. So if you found our solution interesting, Don't hesitate to contact us, and I thank you for your invitation.
No, just some word about Sophie Blondel. She's a scientist at Yposkesi, and she will talk now about gene therapy at this very nice and big company that we have at Genopole. Thanks, Sophie.
Thank you for this introduction. Thank you for Genopole for organizing this webinar. It's the opportunity for us to present Yposkesi. So I will start with a presentation of Yposkesi. Yposkesi is a spin-off of Genopole, created in November 26, with the support from AFAM, which is a French Association for Myopathy, and the French public investment, the French Bank for Public Investment. Yposkesi is a contract development and manufacturing organization for the production and development of viral vectors, especially for AAV vectors and LV vectors. The offer we propose to clients are process analytical development, process industrialization, and GMP manufacturing for gene therapy. We have two main platforms, two main systems to produce the viral vector. The first one is in adherent conditions in CF10, and we can multiply the CF10 up to 24 CF10 per batch. And we have also a platform with serum-free suspension process up to 200 liter scale in GMP condition. Yposkesi proposed to their client end-to-end services from process and analytical development to large-scale manufacturing, integrated project management. So we can have a real input in bioprocess development, manufacturing in GMP condition, fill and finish, analytical development, and quality control. We have all the quality control required for the qualification of viral vectors, and also a regulatory support, especially for CMC information for IMPD or IND. As a process development, we can start from very little scale at 100 milliliter, and we can go to 200 liters in a pilot room that mimics what we do in GMP rooms. We have also all the DSP steps from clarification, chromatography, ultrafiltration, and sterile filtration. Our main building is has a surface of 50,000 square in GMP condition, and we are roughly double the surface within 2022 with a new plant, which will be just next to the first one, but they are designed differently. The current one has 4 different suites of production with 2 bioreactors of 200 liter scale and 2 independent fill and finish suites for direct products. The other building we are construct at this moment will have just 2 manufacturing suites, but we will equip it with 2 bioreactors of 1,000 liter scale. So at the end we will have 35 batches per year in the first building and 40 batches in the second building. So I would like to speak with you about the obstacle and development opportunity there is in viral vector production. Obviously, it can't be an exhaustive list, but it's just an insight I would like to share with you. First, we see that there is a real increase of investment in gene therapy. First, there are a lot of actors in gene therapy. It's not just small company, it's also big company like Novartis and Roche. We see also, and Galy already speak about this point, a real increase in clinical trials, and there are very important investments in bioproduction. Several $1 million are on the table now, and it can be more than it, as we see last year with Thermo Fisher or Catalent. So after an effective phase of proof of concept, we observe a dramatic increase in the development of gene therapy treatments. And thanks to this development, we are faced now to an increase of new market authorization of gene therapy on the markets. So there are 2 to 3 new products per year. And we see also that there are around 30 products in phase 3 from different therapeutic areas, from cancer to rare diseases. And it doesn't mention on this slide, but the price of these therapies are very, very high. For Yescarta, it's 350,000 per treatment, but for Zolgensma, it's more than 2 million per patient per treatment. So we can't hear that now, and it's impossible to continue like that if we would like to each patient have access to this kind of new therapeutics. So we have to improve the development again, and to make some effort to decrease this first bottleneck, which is the price. And if we would like to see how to calculate the scale of viral vector we need for one disease per year, you just have to take the dose level needed and to multiply it by the predicted patient number. If you look at an ophthalmic disease, for example, with an AAV treatment, you see that you need 1.5 multiplied by 10 to the 11 VG per eye, and for hemophilia it's more than 10 to the 15 VG per patient. If you multiply that for rare diseases with 10 patient per year, it's just 10 to the 10 VG per year, and we can do that, and we do that currently. But for common diseases with more than 100,000 patient per year, you achieve 10 to the 20 VG per year to produce. And this amount of viral vector, we can't do this now. We don't know how to produce this. So that's why when you look at this chart, which show the near-term manufacturing capacity versus the demand, you see that the demand of viral vector is surpassing the manufacturing capacity in 2020, and the discrepancy will widening over the next few years. So here is my last slide, just to share with you some elements to think about how to improve the productivity, how to reduce the cost of the production to make access to this kind of product. The first thing we have to focus on is lower the cost of goods. The second is shorten the timelines and reduce the lead times. The third is increase the robustness and reduce the cost of poor quality. And the last is increase the flexibility of our platform. So the facility of tomorrow will have to implement several changes. First, from starting materials with new plasmid construct, new platform, new cell platform like stable producer cell lines to reduce the amount of plasmid, which is a huge part of the price of the cost of production. But if we would like to go further with real breakthrough innovation, we have to think about new expression systems to produce the vectors. We have to focus also on USP steps with process intensification, scale-up systems, continuous bioprocessing for antivirus, optimization of the transfection with new agent and new condition and so on. This list is not exhaustive. It's impossible to be exhaustive in 10 minutes, but it's just, again, some insight to share with you. And in the DSP, we have to do some effort too, especially to increase the purity of the product with an enrichment of full capsid and the implementation of novel affinity chromatography matrix. And I will finish with the quality control, because we would like to continue to produce therapeutical treatment with an important quality, and we have to focus on better characterization of process-related impurities. We have to enhance inline monitoring, automatize the methods, and implement multivariate analysis and predictive modeling. And I can't finish my presentation without speaking about patients, and that's why we are working now with AFM Téléthon, and with the aim to improve the daily lives of patients by promoting better access to care and better social support.
Well, good morning to our colleagues from the United States. So where is the click? Yes, here we are. So thank you for this opportunity to discuss about strategy for safety of cell therapy products, an important matter, I think. But rapidly to introduce Texcell, who we are, Texcell, and what are we doing. We are in business since 1987 as a spin-off of the Pasteur Institute in Paris, and we moved down to Genopole, Évry in 2005, expanding our labs. We had a substantial growth over the years, both organic growth and both by acquisition, and I want significantly to mention here Texcell North America, which is based in Frederick in Maryland. So Texcell is also present in Germany, in China, in Russia, and Japan. Texcell, we are a specialist in viral safety. That's our core business. That's our history of Texcell, viral clearance. But we also have an immunology platform for custom testing. We have a in vivo testing on small animals. And we also have two manufacturing suites for GMP cell banking, but for API and not finished product, on the contrary to Yposkesi. So let's dive into the topic. So as was said before, there are two types of cell therapies, autologous and allogenic cell therapies. I would like to point out, though, that this is not an entirely new field of activities, as this practice is well established for decades, at least in the principle. And I'm thinking here of transfusion medicines, of course, which is the oldest of cell therapies. Of course, with the recent developments of the genetic technologies, this field of interest is ramping up very, very fast. From the beginning, there has been safety issues. First, immunological reaction were the first to be addressed, and we have heard earlier this morning that yes, immunological reaction to vectors is an important thing. But there's more to that, and what are those risks? So, the risks identified today are, as one can understand, a sum of knowledge accumulated over the years and summarized in this single slide. From that slide, I would like to group the risks into three sections. Firstly, there are risks inherent to the cell themselves. Either the origin of the cells, are we an autologous system or allogenic system, on the carcinogenic potential of the cells, the ability to proliferate or to differentiate for the cells, or as brilliantly stated before, the immunological response, the ability to initiate an immune response into the patients. That's one thing. Which leads us to the second group, the manipulation and manufacturing of process of the cells. We talk a lot about genetic manipulation, but there's also differentiation of the cells depending on the program you're doing, expansion of the cell, all steps of the manufacturing process that have inherent risks. The reagents used on the cell culture are potential source of contamination, as is a process or operator a source of risk, despite all the stringent precautionary measures taken. At last, the mode of administration is to be considered too, in particular when there is an external material as vehicle, but we can mitigate these risks with a proper safety strategy. Let's first say that there is no one-size-fits-all strategy. Each project is a particular one, and testing have to be adapted. So, in risk mitigation, there are 6 points I would like to draw your attention to, 6 points to be considered. The first one is the identity and purity. What are you injecting exactly? What type of cells? This is especially true for cells who could or would undergo differentiation, like stem cells, for instance. So there are, you see in the slide, some technologies that can be used to address those risks and to give an answer for the authority and for the safety of the patients. There is also the second group would be the cell stability. Both cellular and genomic stability have to be tested. For instance, let's mention in the karyotyping, for instance, the stability of the insert, the copy number of the insert, the flanking regions, etc., in case of GMO cells, of course. Most important and recognized for long is the bacteriological status of the cells. The manufacturing process is, despite all the precautions, a potential source of contamination. So we have to test for adventitious agents, which is the canonical term for viruses, bacteria, fungi that can be, and viruses, of course, that can be in the cells. Let's mention some things. For instance, the source material. If you use FBS, for instance, a fetal bovine serum in the process, well, you can have viruses, bovine viruses, that can be transmitted to humans. The same could be also true for using a very widespread trypsin for passing the cells, even though there are no recombinant ones. Generally speaking, all adventitious agents, which mean viruses, should be looked for. So the next group is tumorigenicity. Tumorigenicity is important to consider. And of course, again, this is very dependent on the cell types that would be used in the therapy. Biodistribution. Biodistribution is important as well. One wants to be sure that the injected cells won't be crawling all over the patient after. So there are means to check that, of course. And at last, toxicology, especially, and I would mention here because this is sometimes forgotten, especially when there is a technological vehicle in the administration process. For instance, we work at Texcell with different companies that will encapsulate the cells before administration to the patient, and of course the toxicology of the product should be tested as well. Finally, and that will be my last slide here, one point to consider in the strategy is the balance between time and safety. Nucleic acid testing may be fast, but you detect only what you look for, obviously. Or if you have a too sensitive method like the NGS, for instance, it may raise more questions than give answers. On the other hand, tests on living cells or animal can take longer to give results, but they have a broad spectrum, and they also work for poorly characterized agents, not to mention emerging viruses. Thank you.
So thank you, everybody. So we have one question about IP between Asia and Europe for gene therapy. It's a huge question. I don't know if we have any answer in my with the speaker, I don't think so, about the difference between IP in Asia and Europe for gene therapy. Yeah, no, no. In fact, yeah, I think it's a huge debate. So we in fact don't know. And to be clear, we of course can come back to our IP expert, but currently we don't have answer. I will try to get it and to send you any answer. So anyway, about the rest, what I can say for first of all, thank you very much to our speakers today. And thank you to be part of this seminar. We will send you to everybody. We'll send you the presentation, and you will receive a special link, a personal link for the YouTube presentation. What else I can tell you? You still can contact us for any kind of question or demand or request about Genopole, Paris region, Yposkesi, Texcell, Genopole, White Lab Genomics, and Galy and everybody. We are here to help and to increase science and business in gene therapy in France. We try to be at the middle of this big discussion and big market. So thank you very much. What else I can say? Thank you. It has been nice to have you tonight, and bye-bye.